So vegetable grafting, I have seen the last year JRF question paper. There is a question on vegetable grafting. Vegetable grafting is done against what? Soil-borne diseases or airborne diseases or something like that. Options are there. So I have included here the vegetable grafting. What are the purposes for the vegetable grafting? When you use a rootstock, you all know grafting. How do we do grafting? There are two questions in grafting. There is a rootstock and there is a scion. We join the scion with the rootstock. Rootstock is the lower portion, it produces roots into the ground. Where the scion is the above portion, it produces the flowers and fruits, right? So the rootstock effect is there. When during grafting, you can improve the yield and quality. Quality means improving in the nutrient contents, protein content, and new vitamin content. So the rootstock effect is there on the scion in increasing the yield and quality. We will see the examples. Stress resistance. What type of diseases can we solve through grafting? The diseases are the soil-borne diseases, because here the rootstock is the resistant one. Rootstock is the resistant one, and rootstock produces roots, and rootstock is connected to the soil. So when you use a disease-resistant rootstock and a susceptible high-yielding scion, you can produce high yield without the problem of the disease. The scion is high-yielding type but susceptible to disease, but the rootstock is a wild type but it is resistant to disease. So you graft it. Take the rootstock from the wild and scions from the cultivated high-yielding type. You can get higher yield without much loss due to disease. So soil grafting is very effective against soil-borne diseases like fusarium wilt and bacteria. This was the question last time. Grafting was good against which type of diseases? Soil-borne diseases. Examples: fusarium wilt and bacterial wilt. At the same time, grafting is also useful against abiotic stresses like low temperature, heat, drought, flooding. It's actually these three: drought, flooding, and salinity occurs in the soil, isn't it? Drought, flood, and salinity. All these three conditions are related to the soil condition. So that when the rootstock is tolerant to these conditions, you can grow you can graft the susceptible scion to the tolerant rootstock, and you can get good higher yields without the problem of the stress. And inducing flowering. Grafting can be used to induce the flower. So sweet potato cultivar, this I will talk in next slide. So sex modification, this also I will talk in next slide. Sex modification means generally in case of monoecious types, in dioecious types, male and female plants are different. In case of speed gourd, spine gourd, and pointed gourd, 10% male plants we have to plant in the field to induce flowering, isn't it? So these 10% plants are waste. They occupy the land, but they do not produce any fruits. These 10% male plants. But what happens when you graft a female scion onto this male parent? It produces the lower portion of the stalk produces the male flowers, and the female scion produces the female flowers. So that you can convert this unproductive 10% male plants also into productive. So here are the examples. Yield improvement was done in potato scion Kufri Sinduri and the tomato rootstock Pusa Ruby. Yield increased in the scion cultivar. Oh, sorry, here is the mistake. Excuse me. The rootstock is potato and the scion is tomato. I'm very sorry, I didn't see it earlier. So yeah, disease resistance. The rootstock, disease-resistant rootstocks are in brinjal, Solanum integrifolium and Solanum torvum. These two are the wild species. Solanum integrifolium, a wild species resistant to fusarium wilt. Solanum torvum, wild species resistant to bacterial wilt. When you use these two wild species as the rootstocks, we can overcome the fusarium wilt and bacterial wilt in brinjal. Abiotic stress resistance also, by using the raw rootstocks like Solanum melanogenea and Solanum habrocheates, we can overcome the stress problems. And quality improvement. In case of tomato, a hybrid rootstock, Solanum lycopersicum and Solanum cheesemani, this hybrid rootstock can be used in tomato for improving the quality, like total acidity and TSS content. And flower induction, especially these tropical tuber crops, when flowering is very problem in tropical tuber crops. So by grafting onto a flowering cultivar inducer, we can induce flowering also in non-flowering types. And as I told earlier, modification of sex form in Momodica cochinchiensis, By inducing the female flowers on male plants, by grafting the female scion, we can convert the 10% unproductive male plants to also female plants. So just remember the exams. You just the brinjal rootstocks are very important. Solanum integrifolium and Solanum torvum. Solanum integrifolium rootstock resistance against fusarium wilt, and Solanum torvum resistance against bacterial wilt. These rootstocks are very important. Classic examples they ask. I think in IRI PhD examination we have asked this question. So just I will give very brief about biotechnology. Very brief. So meristem culture generally we go for the elimination of viruses. So in onion, shallot, pea and brinjal for the elimination of different viruses, meristem culture is followed. Anther culture for the development of homozygous lines. So from anthers are N, not 2N, not N, pollen grains. Pollen grains are N, not 2N haploids. So when you grow the pollen grains in a culture medium, they produce haploid plants, and to make them diploid, you have to double the chromosomes by giving the calcium treatment, so that 100% homozygous plants we can get from the anther culture. As I told earlier, in chili, in capsicum frutescens, Haihua 3 variety was developed for anther culture. Embryo rescue, when in distant hybridization, when two species are crossed, there is generally a failure of the cross due to hybridization barriers. In case of if the hybridization barrier is embryo abortion, we can take out the immature embryo and we can culture them on the tissue culture media, and we can grow it into a plantlet. So these are the process where embryo rescue is used. Somatic hybridization. Somatic hybridization is different here. Two different cells we put in a test tube, we add a fusogen, and we fuse two cells of two different varieties or two different species. So here the cells of two different species, Solana melangina and integrifolium melangina and Saint Concessive, are fused for obtaining the resistant pseudomonas. So here the important thing is broccoli to cauliflower, transfer of CMS ugra. In my next class, I will talk about this process. But remember this one: broccoli to cauliflower, the cytoplasmic male sterility was transferred through somatic hybridization. In potato also, in tomato also. So transgenic viral resistance. So engineering, through genetic engineering, we have developed the varieties that are resistant to viral diseases. The transgenic product is mostly viral coat protein. Like this viral coat protein gene, when you introduce this viral coat protein gene into a plant, host plant resistance will be there. So the binding sites will be blocked by this viral coat protein, so that when the actual virus comes, there will not be any binding site. For more information, you just read the book. Now we don't have much time, so this concept I cannot explain right now. You can contact me for any clarification regarding the concepts. So these are the examples where the vegetables are engineered for viral resistance, and likewise for fungal resistance also. These genes, chitinase, the mechanism of fungal resistance is degradation of fungal cell wall. We all know that fungal cell wall is made up of chitin, isn't it? So the fungal cell wall is made up of chitin. So when you transfer this chitinase gene, it will degrade the fungal cell wall. Automatically, the fungus will die. Likewise, osmotin. Osmotin means it will make the fungus dehydrate. Fungus will not get much water. Likewise. So these are the examples. Bacterial resistance also. The BT insect also. The BT insect again from the bacteria, soil-borne bacteria, Bacillus thuringiensis. So you all know what is BT, how it works. No? So the mechanism I don't go much deeper. So the BT vegetables are developed here. Cry1AB against Helicobacter armigera in tomato, Cry1H in sweet corn against same Helicobacter armigera, Cry3A against Leptinotarsa disseminata. No, the cabbage, diamond backmark, Prutella gelastella, and against cabbage, butterfly. So these are the BT genes transferred into vegetables against various insect resistance. So what is synthetic BT? So this GY1AB is similar to Cry1AB, but this is not directly taken from the bacteria. The sequence of the gene synthesized in the lab. So this is synthetic BT. The gene sequence is same, but the gene sequence is not taken from the bacteria. The gene sequence is artificially synthesized in the laboratory. That is something which is nothing but. So in brinjal, tomato, cabbage, but unfortunately none of these transgenic varieties are allowed for cultivation in India. As I told earlier, the BT brinjal was allowed for commercial cultivation in Bangladesh 4 to 5 years ago. They have been successfully cultivating BT brinjal and harvesting, and farmers are having good profit. So far, no side effect, adverse effect on human health or environment has been reported. It may be there in the long time, may not be in short time, but in long time. Actually, this BT is always a very debatable topic. Some people are opposing, some people support that. Until and unless there is a strong scientific evidence, we cannot support anything. So gene transferred other than BT, these are the genes from some anti-nutrient factors from the other crops transferred to the different crops. These anti-nutrient factors also sometimes they uh, provide resistance against insects. That is we call antibiosis. For example, gossipol content in cotton is provide repel the insects. Likewise, the sinigreen content in the brassica vegetables, it repel the insects, the diamondback moth. So the anti-nutrient and alkalines present in the plants, they repel the insects. That is called antibiosis. So this antibiosis genes also transferred to the one crop to another crop for providing resistance. So the proteinase inhibitor, trypsin inhibitor. So when the insect eats, the insect will not get protein. Proteinase inhibitor, the insect will not get protein. Likewise. So all trypsin inhibitor also protease inhibitor. So engineering for herbicide tolerance, why it is needed? So generally, when the herbicide is sprayed, it kills the crop plant also. Along with the weed, it kills the crop plant also. So when the crop is resistant to herbicide, the crop will not die, only the weed will die. So these are the herbicide resistant genes that are transferred to various crops against various herbicides like atrogen, bromoxyl, and phosphino, L-phosphinothrixin. So engineering for post-harvest rice, especially in tomato, that long, as I told earlier, these fruits and vegetables have very less shelf life. So we have to increase the shelf life of these fruits and vegetables. So how we can increase it? So we by slowing the ripening, by slowing the ripening process, we can increase the shelf life of the vegetables. So all these genes, antisense, polyglutamate, ACC synthase, and ACC oxidase synthase, phytosynthase genes. These genes are these enzymes. Actually, ACC synthase and ACC oxidase are enzymes in the ethylene pathway. So ethylene is a ripening hormone. Ethylene is responsible for the ripening. So these genes are these enzymes are in the ethylene biosynthesis pathway. So by blocking the genes of ACC synthase and ACC oxidase, we can block the ethylene production, and we can slow the ripening, and we can increase the shelf life. So this is the very important slide. Those are the concepts only. What are the varieties developed by the private sector in India by transgenic means? Not only in India, all the worldwide. So flavor saver, you know that antisense technology they used for increasing the shelf life in tomato, like Colgene company, and for quality improving in tomato, the endless summer variety. And Colorado potato beetle resistance in potato, the variety is new leaf. And same Colorado potato beetle resistance in potato black jacket, and viral resistance in Splash Freedom 2. This Freedom 2 was the previous question. What is the viral resistant Splash transgenic variety Freedom 2? So just go through this slide. And thank you for your patience listening. I think I might have possibly boring.